Imagine that you are a scientist and a blueberry farmer who notices that some of your blueberries have really firm skin while others are really soft. You do some research and find out that a protein from a certain gene A is responsible for the softness trait in blueberries. You also discover that a protein from a certain gene B is responsible for firmness. You want to figure out if there is a way of not producing or producing very little of this protein from the gene A in the eukaryotic blueberry cells. While referencing literature, you are unable to find any blueberry cell lines or strains that have mutations in gene A. You must take it upon yourself to solve this issue. In order to genetically edit the blueberry plant so that they do not have soft skin, you can either use CRISPR or RNAi methods. CRISPR is a molecular gene editing technique derived from a bacterial antiviral defense mechanism. CRISPR locates and cleaves genome target sequences with the help of the bacterial protein Cas9 and a site-specific guide RNA. Together, the CRISPR-Cas9 editing system results in a site-specific double-stranded break in the targeted DNA, resulting in the deletion or insertion of a specific segment of a gene. RNAi is a cellular mechanism that uses the gene's own DNA sequence to turn off or silence the gene itself. Long double-stranded RNA is cut into small fragments about 21 nucleotides long by the dicer enzyme. These fragments are referred to as siRNA and bind to proteins from the Argonaute protein family. After binding, one strand of the double-stranded RNA is removed and the other is available to bind to the messenger RNA. Once bound, the Argonaute protein can cleave the messenger RNA and silence it. When comparing and contrasting, RNAi and CRISPR are both methods of gene editing, but have very different mechanisms. CRISPR is derived from bacteria, while RNAi is derived from RNA. CRISPR uses Cas protein, while RNAi uses Argonaute protein. CRISPR is involved in the knockout of genes, and RNAi is involved in the knockdown. CRISPR is applicable at the DNA level, and RNAi is applicable at the RNA level. CRISPR silences genes permanently, and RNAi temporarily. Lastly, CRISPR has low off-target effects, and RNAi has high. Both methods could be used to increase firmness in the blueberry genome. However, if I were the scientist in question, I would use CRISPR-Cas9 methods to completely knock out gene A. Furthermore, CRISPR-Cas9 can be used to replace gene A with gene B. I think that these efforts can be used to increase firmness in the blueberry genome.